Well, welcome at this course, and thanks for giving me the opportunity to present here. I think it's really important that academic interest is increasing for cultured meat and plant-based meat, because as we kind of all assume, it's an important area for the future to get our agriculture kind of in a sustainable way and to feed people. I'm not going to talk too much. Actually, Tom has asked me to talk specifically about the fat aspect. I assume in the rest of the course you have covered some of the other aspects of cultured meat, but we are, in addition to creating meat tissue, also creating fat tissue for different purposes, which I will allude to. And he also asked me to talk about ideas for scaling up. So I'm not going to, I assume that you all have heard a lot about why we're doing this, what the detrimental effects of agriculture are on lives of animals and on especially the environment, but also food security. So I'm not going to dwell too much on that and dive into really the technology. But the fact of the matter is I could not resist showing this picture for an Israeli public. You know, feeding the world is a complex issue, and it's getting more and more complex when we go to 10 billion people who have more and more sophisticated needs. If you think about it, our choices in food has always expanded, at least during the last couple of decades. And it would be hard to accept that in the future we may not have all those opportunities. So, but in order to retain those opportunities, we need to seriously rethink how we're going to produce some agricultural products, and in particular animal proteins. Now, if you think about culturing meat, initially, so this is, I hope, a image that you're familiar with. On the upper left hand, a steak, and then if you zoom into the different structures, through muscle bundles, muscle fibers, individual muscle cells, which are not really actually individual muscle cells, and then down to the proteins that we are, as a kind of nutritional value, interested in. You can think of ways to redesign the manufacturing of that muscle, you know, instead of going through an entire animal, to kind of go down that chain and find ways to find substitutes. So you could do this with proteins alone. You can use single cell protein production and have actin and myosin made and have kind of the same nutritional value and maybe some of the functionality as well. But it will be a protein-based system. Some companies, I'm sure, are working on this. The next level is cell-based, where you basically use muscle cells or fat cells and mix them in an otherwise plant-based product, and that you would call cell-based. And I know the term cell-based now has been more broadly used, much to my dismay, I must say, because in the end, every meat is cell-based. But you know, that's a technical issue. I don't want to dwell on that too much. But if you call something cell-based, that opens up the opportunity to have an otherwise plant-based product mixed with cells.
Please mute yourself if you are in the background. We appreciate you joining, but please have yourself muted. Thank you.
Right. Then the next step up is fiber-based, which is basically a muscle fiber consisting of like hundreds of cells that are merged into one functional muscle fiber with all the protein qualities and with the functionality of those proteins. In fact, not only in terms of food functionality, but also in terms of physiological functionality. These muscles actually contract, and the fat cells obviously not, but they have a similar kind of function. And then if you move further up, you go to tissue-based products, basically what Aleph is doing in Israel, using more cell types and using a scaffold to build an entire tissue from the outset. And that I would call kind of tissue-based, although that name is not en vogue currently. That for most of the other companies is also a final goal, but some companies like ours and some of activities like ours kind of focus on this more fiber-based intermediate because it's just easier. Okay, so the process is pretty simple in a larger kind of aggregation level. You start with a biopsy from an animal. At least in our case, I will come back to that later. You start with a biopsy from an animal, then you isolate the cells and you select the cells. You either muscle cells and fat cells or just muscle cells, and then most importantly, you let them proliferate for many doublings, as much as you can. And that's important because that is basically underlies the entire technology. By multiplication, you can scale down the number of animals that you need for meat production and thereby gain kind of the benefits that we are all aiming for. And then finally, in our case, that's the final step, you differentiate them into muscle fibers and fat cells. And then, of course, you assemble it into a hamburger. Now, except for the fat cells, this is what we did in 2013, made basically 10,000 of these muscle fibers and combined them into a patty. This is how these muscle fibers look under the microscope. It's basically a large number of cells. Most of them have merged into a muscle fiber and they start to contract and they start to become a real muscle fiber. And as I mentioned, in 2013, we took basically that process, which already existed for about 15 years in the medical field, just made 10,000 of these fibers, which was very tedious work, and made a hamburger out of that. And as you may recall, that was kind of internationally televised in a sort of hybrid between a cooking show and a press conference. And that, in retrospectively, I was very happy, and I'm still very happy with that event, because that basically sparked a lot of interest in the world to start doing this, which now has accumulated into like a couple of dozens of companies pursuing this. The interesting part of that presentation was that it kind of looked like a product launch, because we had to cook it and have people eat it and comment on it. And I tried to avoid that, but it was basically, it ended up looking like a product launch. And we knew, of course, that it wasn't a product yet, because there were still a lot of issues with it in terms of still animal components in there, the way we grew things, the way we kind of expanded it. So there were still issues, which I tried to illustrate by the fact that, and we specifically mentioned this, that this hamburger, this single hamburger, cost us €250,000 to make. So I was hoping that everybody would realize after that, well, you still need to do quite some work to get where you eventually want to be. All right, so how do you go from there to a product? And of course, the production has to be efficient. That's why we're doing this, right? To reduce the impact on the environment. So for that, it has to be efficient. Sustainable, meaning that every component that's in there is of indefinite or seemingly indefinite supply, because you can basically source it or recycle it. And it has to be meat, because people, at least 95% of the population, not the population, but the people in Israel or Europe or the US that are not vegetarian, like the meat product. So usually I go in kind of the order of efficiency, sustainability, and mimicry, but because of the focus on fat, I switched to mimicry immediately. And mimicry is, well, there are a couple of components of meat. Of course, there's the texture, there's the color, and obviously there is the taste. And as everybody probably realizes, who has tried, even in blind tasting at some point, different types of meat, the fat is really an important component of the taste. It's not the only component, but it's really important. If you sample a very lean piece of lamb, you will have, and you do that blindly, and trust me, I've done that, and I have failed pathetically to recognize it as lamb. It's without the, but if there is a sliver of fat, of lamb fat on that lamb chop, you immediately recognize it as lamb. So the fat is an important component of the taste. It's also a nutritional value, and not only a positive nutritional value, it can also be a negative nutritional value, because we know that especially beef and pork contain very, very small amounts of polyunsaturated fatty acids. So it's mostly saturated fatty acids that increase your cholesterol and eventually lead to increased cardiovascular disease. And that's why the World Health Organization, that's one of the reasons why the World Health Organization has recommended that you should not eat more than 300 grams of red meat per week, per person, which is really not a lot. Now, having said that, the relationship between polyunsaturated, or the relationship between saturated fatty acids and cholesterol is relatively clear. The relationship with cardiovascular disease at a population level, epidemiologically, is pretty clear. At an individual level, is not so clear. And the reason why I have here the Atkins diet is because you may recall that at some point, like 20, 30 years ago, Atkins started this diet where you basically eat mostly proteins and fats and no carbohydrates, or very little carbohydrates. And everybody would have thought that these people would die like flies because of the increased cholesterol levels. And that was not quite the case. So our understanding of this is not complete, but for the moment, we still are working on the general assumption that if you have saturated fatty acids in your diet, your cardiovascular risk is increased. Then about the taste. The taste is very interesting, and a lot of people have written about this, but it's also somewhat mysterious because it's very complex. The fact that it is so complex is illustrated by that to date, all the tasting analysis is still done by people, by tasters in tasting panels. We don't have really a good equivalent for it. One of the factors, especially for fatty acids, is a receptor CD36, which seems to be important for taste kind of development in taste buds. And of course, you can look at fatty acids that bind to the CD36 as kind of a surrogate for the tasting that you eventually want to do. That works to some degree. If you see here, some fatty acids bind to that CD36 and cluster that CD36 very well. Cholesterol almost does not. So in meat, the assumption is that cholesterol does not really add so much to the taste. And here, surprisingly, some of the unsaturated fatty acids actually are kind of an important taste driver, although in panels, people usually associate the presence of linoleic acid and well, these two gives kind of a fishy taste to it. To increase the complexity of the need for fat in the muscle, this is an older study where You have on the sort of x-axis on the left side non-marbled, so meat that it basically is very lean and has no very fine grains of fat in it, and on the right hand marbled, and on the lower, on the top half a lean meat, and on the bottom half a fat meat. And you see here the different geographies where so there's not a uniform preference for marbling, non-marbling, and fat or lean. It's very geographically determined, which kind of illustrates the subjectivity of this tasting and this preference for fat. From a tissue engineering perspective, it's also a kind of complex item because we have different types of fat. Traditionally, we have white fat, which is on the far left here. These are fat cells. If you take a sample from your skin, you put it under the microscope after some staining, you will see this. These are very, very big cells, like 100 microns in diameter, and they are chock full of fat. And the nucleus is kind of squeezed to a far end of the margin of that fat bubble. That's white fat. That is a storage of fat, energy storage, a metabolic kind of piece of equipment, if you like, tissue. Then we have brown fat, which is quite the opposite. It looks very different. It's kind of multi-locular. The cells are much smaller. It's multi-locular fat deposition in these cells. And this is, we still as adults have some of that brown fat tissue somewhere in the thorax. It's very small. And this is actually not an energy storage. This is an energy consumption tissue. And it basically has, you find this in youngborns, in neonates a lot, and it drives their heat production. So it's one of the systems that is involved in maintaining our body temperature. And that's brown fat. And then you have a third component, well, you actually have four components, and that is very confusing. And that is sort of fatty degeneration. When cells become sick in the body, they start to store fat. So it's very easy in a tissue engineering perspective or in a cell culture perspective to confuse this fat storage as a part of a degenerative process, if you like, with true what we call adipogenesis, maturation into a fat cell. So to give a little bit of perspective, there are some pathways that have been described that drive the fat stem cell, if you like. I should have said it, I think. Fat tissue contains specific stem cells. And they come under different names: mesenchymal stem cells, stromovascular cells, or adipose tissue-derived stem cells. But the fact is they are stem cells. They can proliferate just like the muscle cells, and they can differentiate into fat. Now, these cells are a little bit less designated than the muscle stem cells. They can not only become fat, but they can also become bone or cartilage or fibrous tissue. So in order to drive them through a adipogenic pathway, meaning towards mature adipocytes, you have to kind of stimulate them in a specific way. And here you see that there are actually some ideas on how you drive them into the white adipogenic or the brown adipogenic or something in between, which is kind of called beige adipocyte cell line. And you see that there are some signal molecules that you can use to go from the kind of common precursor to any of these cell types. I can tell you right from the start that in cell culture and tissue engineering, this is still not an easy task. It's really requires some R&D. And to make it a little bit more complete, but also more complex, is that this is just a kind of an extension of that cell lineage thinking. So you start with MSCs, the mesenchymal stem cells that you can get out of that fat tissue or out of bone marrow. And then you can separate them into 3 kind of categories, probably more, but this is limited to 3, based on surface markers, and MIF5 is one of them. And then you see all these kind of downstream transcription pathways driving them into a more mature adipocyte. And what is noticeable is that PPAR gamma and CEPB alpha are kind of anywhere more or less upstream or very downstream in that pathway. Those are the 2 transcription mechanisms, so CEPB alpha and PPAR gamma, that are kind of the final transcription pathway towards an adipogenic cell line. So somewhere in that differentiation, you have to stimulate those 2 transcription pathways. In tissue engineering, there are a couple of other cues that you can use other than the biochemistry cues. You can look at stiffness of the substrate on which you grow them, for instance. So if you have a very, very sloppy substrate, like with an elasticity of, or a stiffness of less than 3 kilopascal, you drive your stem cells more into an adipogenic cell line. If you increase the stiffness, they go more towards a chondrogenic or even an osteogenic cell line. And the same is true whether you compress them, whether you use kind of forces like shear force or compression force. You will either drive them more into the chondrogenic or into the adipogenic cell line. So compression typically goes more towards the chondrogenic line, and very sloppy substrates more to the adipogenic cell line. So you can use those to drive your cell into a particular line. Now, differentiating mesenchymal stem cells to fat cells has been done for decades. And in fact, it's one of the definitions of a mesenchymal stem cell that you can drive them into either adipogenic cell line, chondrogenic cell line, or osteogenic cell line. And the protocol to make them into adipocytes actually is very well described, but it consists, unfortunately, of a number of factors that are very cumbersome in food production. One is insulin, which is fine, I guess. 2 is dexamethasone, which is a steroid, actually a very stable steroid, and we don't want to associate steroids with our food production, especially not the synthetic ones. The third is IBMX. IBMX is a super caffeine, which is toxic for people. So you don't want that in your food either. And then the fourth is indomethacin, at least in some of those stimulation protocols. But those are kind of necessary or deemed necessary to stimulate that CEPB alpha or PPAR gamma pathway, or the combination of the 2. So the challenge was to basically design a system that was compatible with food and doesn't have kind of those negative associations with IBMX and high levels of steroids. We are focusing on beef, so we're also focusing on bovine fat tissue, and it's a choice. We have lots of reasons for that choice, but the only thing I want to say about this is that everything I'm saying, or everything what we are doing, is rather specific for bovine fat tissue. That doesn't mean that for other tissues it's completely different, but you have to optimize it for every species and for every type of cell. And that's basically the challenge that all these companies are facing. You just cannot translate a protocol simply from one species to another. And in fact, you know, the taste, but also the texture of fat is very different between species, so that there is a lot of the similarities in the muscle is much bigger than in the fat tissue. In the fat tissue, there is really a lot of difference in fatty acid composition, in texture of the fat tissue, but also in the way you drive the adipogenic process. So we started with just harvesting fat tissue from an animal, usually after a slaughter, and then we developed an entire protocol on how to get our adipose tissue-derived stem cells or mesenchymal stem cells out of them. It's published, the methodology. You can look it up. And then we thought, well, you know, what drives these PPAR gamma and CPB alpha transcription factors? And basically the typical agonists for PPAR gamma are basically fatty acids. So we tried every fatty acids that's known to mankind and that we had available in the lab, and threw them on these cells and see whether they could become fat cells. So these are just a couple of examples where we have phytanic acid and pristanic acid, and it doesn't really matter. These are just regular, naturally occurring fatty acids. And see whether we could make these adipogenic. Now, these images are 2D cell cultures, which is kind of the basic model system for creating, for kind of optimizing this adipogenesis. It's not the ideal culture system, but it works, and for screening, it's fine. You see that these look more like brown fat cells. They have these multilocular fat droplets. Every red droplet here it's stained with oil red O. So every red droplet is a fat droplet. The good news is that they do take up fat. The bad news, if you like, is that it's still kind of brown fat tissue and not the real white tissue that you want to get. And the question is whether this is true adipogenesis or this fatty degeneration that I mentioned. So you can follow up with a couple of specific markers for fat, like PLN1 or PREF1 or lipoprotein lipase, or a lipase, sorry, or FABP4. There are all these markers specific for fat cells that you can test. And obviously we have done that. Now, 2D is fine as a screening method, but one of the problems with fat tissue is, or with fat cells, if you're really successful, the cells become buoyant because fat is lighter than water. So they lift off the plate and you lose them. And when they detach from the plate, they basically die. What we started early on is to trap them into a matrix, and we typically use alginate because it's cheap and it's food grade and it's very biocompatible, no problem, and it's actually edible. So we started to use alginate, and because we also had functionalized alginates with small peptides, we try to see whether there's a difference if you just capture them in an alginate and let them mature into fat cells, or if you kind of functionalize that alginate so that there is an interaction between the matrix of the alginate and the fat cells. And here you see some of those results. We have repeated this with many different functionalizations, but it matters a little bit whether you functionalize, but not that much. So our current working hypothesis is, and working system is, that we just encase them in alginate or in whatever matrix, doesn't really matter. But you have to kind of keep them in that. in that system, and then you can culture them for many, many weeks. One interesting study from Japan, from my friend Soji Takeuchi, is where they made a rather complex system where they made a core shell technology where the core is collagen and the shell is alginate, and you have the mesenchymal stem cells in the core, in the collagen core. And I show this, the technology is there, it's relatively simple to do. These are, by the way, human mesenchymal stem cells, and mind you, I said there is a lot of difference between species. But I was, when they first show me these results, especially these big balls of fat at the in those fibers, I was really impressed, and I thought, well, you know, this is real white fat tissue. So we have repeated those experiments in our case with some reluctance, because we don't want to use collagen basically in the whole system. Because collagen is animal based, not necessarily, but currently it's mostly animal based, and we wanted to get rid of all those animal components. So I just want to show you this because this is from our own results. Unfortunately, I cannot disclose entirely how we arrived at these results, but we basically used non-toxic stimulants, think fatty acids, all the components of that metabolic system, in an alginate matrix, and you can get these really, really big adipocytes. And the other good news, which I'm really happy with and proud of, is that almost 100% of the cells actually differentiate into adipocytes, which makes it a very efficient system. So I'm pretty confident we get there. These results are, of course, we show very good results, but it's actually a pretty robust system. So we kind of have a good handle on the predictability of this. What we do know the composition of this in terms of lipidomics. We have done those analyses, and fortunately, it already has kind of serendipitously a high level of polyunsaturated fatty acids. So we don't find it tasting like fish. So that's those are two good parts of it. There are still differences with respect to the composition of beef fat. Specifically, the amount of beef fat is very, very rich in oleic acid. And the oleic acid profile is still a little bit lower than in regular fat of beef. So it's not quite optimal yet, but you know, in terms of, we originally thought we had to kind of manipulate it to get more polyunsaturated fatty acids, but fortunately in our basic system we already have quite a high level of polyunsaturated fatty acids. All right, you could ask, you know, why are you going through all those hoops to make fat tissue? You can just add fat, plant-based fat or whatever. One of the reasons is that we eventually want to make a steak, and that you have to kind of have the tissue there and not kind of non-structured fat in there. So the other reason is kind of depicted here, that we know, we haven't done those experiments ourselves, but there's quite a bit of literature on the interaction between fat and muscle tissue, and they kind of beneficially influence each other. So that's, I won't have time to go into this slide because I realize I'm kind of taking a lot of time. There's one company out there that you may have heard of, which is Cubic. They are going the route of basically producing fat, either fat or fat tissue, for the entire industry. And they're using specific processes for not kind of making trans fats, but making the fats like the right structure without modifying them too much. And they are taking the approach of cultivating fat cells for the industry. Now, the last part I want to spend on scaling up, and that's necessary for efficiency and for cost reduction, and also introduces the element of recycling, which is necessary for cost reduction. I will speed a little bit up. So the process of proliferating cells is in our hands very robust. These are proliferation curves over 40 days of culture, and all the different curves are different animals, so different samples from different animals, and they all look very much the same. They're pretty much run in parallel, so it's a very robust system. And currently, we are at about 33 doublings from this muscle sample. And if you do that calculation, you get kind of the muscle sample is 0.5 gram. So it's a very small sample, and you get kind of 2,000 kilograms out of this muscle sample. So we are kind of happy with that. But obviously, we want to extend that amount of doublings. One of the things that we are specifically using is adult stem cells, and they do have a limitation in how often they can double, which is kind of studied by Hayflick already 60 years ago, or 40 years ago. And he said that adult cells typically can go through 50 doublings if you start young enough. Now, if you go back, that 50 doublings would end up with a tremendous amount of muscle from one cow. So that would, if we reach that, then we are fine. I think we're currently also fine, but if we reach the Hayflick number, this is an exponential curve. So the farther you get, if you get from 33 to 35, you have a 4-fold increase in the number of meat that you get out of a sample. Now, other people in other companies are using more immature cells, typically iPS cells, induced pluripotent stem cells. And obviously they have the advantage, they're easier to grow, they're easier to multiply for a longer time, also not indefinitely, but for a much longer time. But then you have to differentiate them into muscle, and that's where usually the problems kind of start. Maybe you have seen this before, but there's a company out there, and they're serving another company. They're using iPS cells, and basically to use iPS cells, you have to genetically modify them. That's one of the reasons why we have chosen not to do that, because in Europe that will be problematic from a regulatory and a consumer acceptance perspective. And in order to differentiate them, this is on the right-hand side, you have to modify them again. You have to basically put MyoD into those cells to let them differentiate into a muscle cell. So we have chosen not to do that, but it's basically a choice. And I'm not going to tell you which is right or wrong. That's not my place. We have chosen not to do that because we fear that in Europe, GMO is not going to fly well, especially in combination with cultured meat. In order to make that hamburger, we used, this is kind of a astonishing slide, because we used a truckload of plastic to make one hamburger. So it's evident that, in a regular cell culture technology, and this is not even quite regular cell culture technology, will not suffice for scaling up. It's just a no-go from a sustainability point of view. So this is the process that we're looking at currently, and that everybody is looking at. So you start with small samples of muscle cells. You then move gradually up to larger bioreactors, and you grow them eventually in tanks like these 100 or 1,000 liter bioreactor, or maybe even 25,000 liter or 500,000 liter. The industry has a lot of these type of bioreactors. But in order to do that, you have to let the cells grow on microcarriers. These cells need to grow on a surface. So to get them in suspension in a bioreactor tank, you have to provide that surface. And it's typically, microcarriers are used for that. There are small spheres of 100 micron, 150 micron in diameter, and the cells grow on the surface of that sphere. And that's a relatively easy system and known system to transform a cell that is growing on a surface into something that you can scale up in a suspension. If you do all those experiments and those optimizations here on the top right, you see those microspheres with the bright dots on it are all cells. You get an exponential growth curve. And if you retrieve the cells from the microcarriers, because that's obviously what you need to do in the end, because the microcarriers are not part of the food, they still differentiate into muscle fibers. We have characterized and recently published that you can use different microcarriers for that, and they work fine. Again, it's not an ideal system, but it's a system that works for the moment and that can get you to an industrial scale. We grow these cells currently in the absence of serum, so it's a serum-free medium, and we can grow them in the absence of antibiotics, so that's all fine. This is also recently published, and these are recent data where we are kind of have optimized these serum-free media. All the curves are different serum-free media formulations. And in the blue, which is the top one, is serum. So some of them are kind of similar, and they actually follow kind of the same exponential growth curve. So that serum-free issue is solved, if you like. Now, how would you go through kind of an industrial scale? These are some of the numbers that we use in those calculations. These are just projections on how we envision this is going to happen. We are not at that scale yet. This is the number of cells that you have per kilogram of meat, cells that you have per milliliter of fluid in a tank, and then per obviously cube meter, because you have to have large tanks of these, and then you eventually can get to a volumetric productivity of tons per cube meter per year. And depending on the cell density, the low is kind of 4 tons per cube meter per year, and the high is 20. So 1 cube meter bioreactor, which is basically a very small cube meter, you can imagine how much it is, not very big, can actually produce a lot of meat in a year, assuming that you have them in there for only 1 week. But still, if you do that math, the world meat production is tremendous. Here it's 300 million tons per year of meat production. So if you translate that into amount of bioreactors that you would need, it's like 20 million bioreactors of a cube meter. And that's 1.5 the fermentation capacity we currently have. Think fermentation capacity, mostly beer and wine and things like that, but also in the food system for enzymes and in the pharma system for proteins. So it's a huge increase in fermentation capacity that you would need to create this type of meat. If you look at the farm level, so if you scale down a little bit, and assuming that at some point a farmer would think, oh, maybe I can convert my barn into a bioreactor space, then a farmer would need about 4 of these 1,000-liter bioreactors to replace his herd of cattle. So that's kind of an interesting perspective, I think, because that seems to be doable. But then you would have to, you have to scale out instead of kind of scale out to multiple smaller units to get to those 20 million cube meters of bioreactor space in the world. Last thing about price. For scaling up, obviously, you also need to reduce the price. And that this is just a breakdown, projections again on what it would cost if we have recycled, if we have, I will come back to that, if we have recycled, if we have reduced the price of growth factors, which is the biggest cost factor, and if we have scaled up. And then you end up with like 140 euro per kilo, which is still very high, but it's already kind of marketable for restaurants. What we are doing currently in all our experiments is do a metabolic analysis of expenditure of that medium, because what we know is that if we, after 2 or 3 days, we throw away medium, we throw actually away a lot of resources. And so it makes it very expensive, but also extremely inefficient. So here, if you look at the essential amino acids on the left and the non-essential amino acids on the right, you see that even after 7 days of culture, pretty much 50% of all the amino acids are still there. There are a couple that really have been depleted, but the majority are still there. And of the non-essentials, kind of quite predictably, some actually do increase in content. So we have to become much smarter using this medium again, removing the waste components, using this medium again, and optimizing the efficiency. That 140 euro, 138 euro per kilogram does not take into account all the nutrients of the medium. And once the growth factors are no longer a cost factor, the nutrients become a cost factor. This is where GFI actually spend quite a bit of research on, and they have identified this long time ago. What they haven't done recently, maybe, but they haven't done so much, is what if you replace the pharma grade amino acids and the pharma grade glucose that we're currently using by feed grade, animal feed grade, and so make every component of a lower grade still palatable for the cells, but at a much lower cost. And I think that's where the final cost reduction from the 140 to maybe 14 or maybe 5 euro per kilo will come from. So we're currently teaming up with the feed sector to make this happen. Then, of course, this has to be regulated. It's a novel food. In Europe, that means that it has to go through EFSA. This is not GMO, so that's it kind of avoids that panel of EFSA, but it still is a procedure where we have to prove that this is safe and fit for consumption. It takes about 1.5 years, so it's kind of painful, but it's also, I think, important to do because we eventually want to secure that this is absolutely safe for people. So my vision for the future is a very conventional one, that we can still enjoy the foods that we have been enjoying for the last 30, 40 years, including meat, but without the negative consequences for the environment. And I hope to have convinced you that we can get there, although it's still somewhat of a path to get there. So in summary, I think this is worth pursuing. There's a high likelihood of success in my mind. Fat production is necessary for taste and texture, and it's possible with food compatible methods. And I think it's also important to do that in view of eventually growing a steak and not just minced meat. Scale up or scale out is a formidable task, where scale out is probably a better way to do it than scale up. And cost effectiveness is a function of technology, recycling, and feedstock selection. And especially the last one is something that we should be working on right now. And of course, regulatory approval is acquired. I want to end with thanking my team. They have done all the work. This is, we're now with 42 people, amazing, mostly scientists, amazing scientists. And I found out that pretty much all of these problems disappear in half a year or a year if you throw very good people and a lot of money at it. So with that, I would like to conclude and thank you for listening and attending. And I'm happy to entertain questions. I think Tom is moderating the question series.
Mark, thank you so much for this really exciting lecture. I have something like 7 or 8 questions of my own, but sadly there are other questions that I would, maybe I will send you my questions on the email. So the first question from Nama Tamo from the course is there a difference in flavor among among the type of fat cells when you were talking about the white adipose cells and brown adipose cells?
Yeah, there is a difference, and you know, brown fat typically is not eaten, so but the composition is different, and you can see that immediately in the morphology, but also in the structure. Brown fat feels very grainy, very not squishy like regular. So it also has a very different texture. So the brown fat, if you culture fat cells, stem cells, and you want to make them into fat cells, the default is to become brown fat. I don't know exactly why that is, but that's kind of the default. And so you have to pull specific tricks to make them into white fat cells. It can be done, as we've shown and others have shown, but yeah, you have to steer it in that white fat direction. It's a little bit of a time issue too. I think the general consensus is that if you culture your tissues for long enough, weeks, eventually they will become more white fat cells than brown fat cells.
Thank you regarding that. I would ask you a follow-up question. Would you put the beige cells, if I remember correctly, you said that they are somewhere in the middle. Would you put them in the same category as the white or as the brown?
Say it again. I'm sorry, I missed that.
You have the white adipose cells and the brown adipose cells. However, you added there an additional one, which is somewhere in the middle.
Right, the beige ones. Yeah, yeah. We have not specifically looked at those, to be honest, because we're mostly focusing on white adipose. That's our final goal, and not beige. It's not quite clear whether this is kind of an intermediate stage, which is stable. It seems to be to some level. I heard about these just a couple of years ago. I mean, in histology, you would never focus on these type of phenotypes. Brown fat and white fat, yes, but beige, you would not focus on it. So for us, it's kind of just interesting from a research perspective that if it's an intermediate, then you know, in the optimization, we know whether we are steering away from brown to white or not. So that's how we're using it currently.
Oh, thank you about that. Just interests me. I heard you talk about it a while back, and I wanted to know more. So this next question from Moriav, also from the course: can we grow adipose and muscle cells in the same media, mimic the intramuscular fat?
Yeah, that's a good question. That's a typical tissue engineering questions of co-culture. And usually what you do if you co-culture cells and you have to differentiate them. So the differentiation medium for muscle is completely different from the differentiation medium for fat. They're two completely different media. So what you typically do if you co-culture them is you find the cell that differentiates easiest and most stable, which in our case would be the muscle cell. You let them differentiate first. And then you switch the medium to the fat medium, and then differentiate the fat cells. So it's a lot of research that's required to optimize this. It's a common problem in tissue engineering where you use multiple cells, And typically, this is the way you do it. You start with the easiest to differentiate and the stablest to differentiate. And once they are differentiated, you do the second one. And of course, theoretically, this can be done because it happens in our body too, right? But yeah, this is what it requires a research and development, but typically it can be done.
Thank you so much for this insightful answer. Gilad Gong, also from our course, he asked, how can we make a steak from small tissues that grow on microcarriers?
Right. Well, so currently the microcarriers are not edible, right? So we harvest the cells from the microcarriers. The microcarriers may be reused. We have done some experiments on that. And then you start with cells. And then whether you grow the cells in suspension or in 2D or microcarriers doesn't really matter. It's just a way to produce the cells, and then you use the cells in a scaffold with other cells or not to make a steak out of it. Of course, it would be interesting to integrate that system and to grow small microspheres or aggregates of complex nature that you can immediately turn into a meat kind of product, not a steak, but let's say a hamburger. And of course, people are, including ourselves, are thinking about these systems that would be ideal, that you can, in suspension, with a certain type of carrier system or scaffolding system, grow cells immediately into muscle fibers or fat spheres, and use them as ingredients for minced meat. I don't see how you can do that for a steak, though. That would be impossible almost. I would say.
Thank you. Next question, are the cells removed from microcarriers using trypsin? If so, are they using bovine trypsin or a non-animal derived alternative?
We are using a non-animal trypsin, which works very well. It's slightly more expensive than bovine trypsin, but not much more. So eventually we want to get rid of that trypsin as well. There are systems out there, even on microcarriers, that have a surface that is temperature sensitive and that can release the cells if you reduce the temperature. So you would not need the enzymatic digestion. Enzymatic digestion is not really problematic. It's just another extra step, and it adds cost to the whole process. Functionally, it's not problematic, but again, from an industrial perspective, a scale-up perspective, it's somewhat, you would like to avoid it.
Thank you so much. I would mention that this question was given by our new internet G5, Stacy Scalò. Another question is, how can you grow a steak or scale out of 1 cubic meters bioreactors for mincemeat? How will producing a steak scale up or scale out?
Yeah, that's a good question, I guess. And we haven't really given that a lot of thought yet, because we are first focusing on the organoids. And there we have very specific ideas on and actually instruments how to automate that. Of course, that requires a level of automation. If you scale out to a smaller scale, let's say like a farm scale or a microbrewery, you want to make the system pretty foolproof, right? So that there is no transition from, because the big risk, of course, if you grow these cells in a medium without antibiotics, that there is a contamination and then your entire process is ruined. So you want to make the transition from one culture system to another culture system, as you want to move that as little as possible, preferably within one system. Now, how you would do that with a steak, you would have to have some sort of automated 3D printing to print the scaffold and the muscle cells and the fat cells in one system. I guess you can do that by lining up a lot of these 3D printers. There are, I can think of other ways to do that in a much easier way. Again, I cannot really talk too much about that because we haven't really patented anything of this. We haven't even tested it. But I think there are more simple methods to do that. My boundary condition that I have not given up yet is still that if you do that, if you make a tissue like that, and you make it full thickness, and preferably like 2 centimeters thick, you would need a perfusion system. So you would need an active perfusion system for every steak that you make out of that cell populations.